Hello, my name is Vanya Chekla, and along with my teammates Sakar and Gabby, we will be presenting vaccines to you. Vaccines help the immune system defend itself against pathogens and protect you from serious diseases. Today we'll give you an overview of vaccines and the types and functions of each one. This is the history of vaccines. In China, around smallpox, scabs are held to prevent smallpox. And in 1800, Benjamin Waterhouse vaccinated his children against smallpox. And then 1990, scientists studied mRNA vaccines to start strengthen immunity. And then around December 2020, Pfizer and Moderna COVID-19 vaccines received emergency approval. First vaccines administered at Northwestern Medicine.
So there are 4 types of vaccines: live attenuated vaccines, inactivated or killed vaccines, subunit and conjugate vaccines, and toxoid vaccines.
Mr. Shifted. What's up? Yes. No, did I mark him present? Okay, I'll go fix it. Uh-huh. Yes. Yes. Mm-hmm. Yeah.
Live attenuated vaccines are weakening disease-causing virus through a series of cell cultures or animal embryos. When the resulting vaccine virus is given to a human, it won't replicate enough to cause illness, but it will still provoke an immune response and protects against future infection.
So inactivated vaccines are similar to live attenuated vaccines. However, instead of being unable to reproduce due to being weakened, they're destroyed using either heat or chemicals such as formaldehyde, and they're still able to provide your immune response, but it won't be as strong as a live attenuated vaccine. The immunity granted won't be as strong as a live attenuated vaccine, and they still have the DNA required to be in a vaccine and warrant an immune response.
Toxoid vaccines are made by inactivating a toxin that causes the disease symptoms. This can be done using chemicals such as formaldehyde, formalin, or other heating methods. It can be considered a killed or inactivated vaccine, but they have their own category to highlight the fact that they contain a toxin and not a bacteria. Some examples would be the tetanus vaccine, subunit and conjugated vaccines. Conjugate vaccines are made of using pieces from the coats of bacteria.
An example of it is from mycobacterial infection vaccines, and self-limited vaccine use any part of a target pathogen to provoke response from the immune system. An example of it is a cellular pertussis vaccine and influenza vaccine. Steps of vaccination is APC cells they detect vaccine antigen and display signal for T cells. APC cells travel to lymph nodes and naive T cells recognize the signal and activate. Naive B cells, they recognize antigen and activate dividing and producing more B cells. After activation of the immune system, plasma B cells begin producing antibiotics. Antibiotics are released and interact with a wide range of antigens. For attenuated vaccines, killer T cells destroy infected cells.
There are two types of immunity, innate and acquired immunity. Innate immunity is the first line of defense, which is the one that we are born with. Once the pathogens reach this, the white blood cells will neutralize them. It can't target anything specific, it's only like a general form of defense.
Acquired immunity is complementary to innate immunity. It is a specific response to pathogens, and unlike innate immunity, it's developed over time through either vaccines or exposure to illness, and it is also, unlike innate immunity, it works on the cellular level rather than on a bigger level like the entire body. So vaccines work with the immune system, more specifically through the acquired immune system, as the vaccines mimic antigens, and antigens are what the acquired immune system recognizes. And through antigens, the acquired immune system will develop a response specific to the antigen and then it'll remember this response for future infections. And the innate immune system can also work with the acquired immune system by alerting it through various means.
Vaccines of the immune system. After receiving the signal from initiate immune system, helper T cells will activate other immune cells, activating and naive B cells, beginning to divide and produce more B cells. New B cells will mature into plasma B cells and will not produce antibiotics for the perceived threat. Antibiotics are released and interact with a wide range of antigens. Killer T cells will kill any infected cells, attenuated vaccines, and memory B cells and memory T cells are going to be reproduced.
So live attenuated vaccines, inactivated or killed vaccines, and subunit/conjugate vaccines can all target both viruses and bacteria. bacterial infections, whereas toxoid vaccines target specifically bacterial infections. Well, the toxins that bacteria. So the bacterial meningitis vaccine is important for college students, as in a small dorms, infections tend to spread very quickly, and because bacterial meningitis is already extremely contagious, it's further amplified by that. And also, by getting vaccinated, it can help prevent future, more fatal side effects from the infection.
Doctors can determine if you need additional vaccine doses based on whether or not the antigen in the vaccine is alive. Live attenuated vaccines can provide a lasting protection with only 2 doses, while other vaccines like inactivated vaccines may require 3 doses to be effective.
More individuals are immune decrease the incidence of the disease and the occurrence of the pathogen. With greater numbers immunized, it is less likely that an immunized person will encounter the pathogen. Mass vaccination confers indirect protection for those who do not receive the vaccine, resulting in herd immunity.
So for the laboratory process of manufacturing vaccines, for live attenuated vaccines, virus will be passed through multiple times through a specific type of tissue in order to better make it suited to that specific type of tissue rather than ourselves. So it'll be weakened to the point that it won't be able to reproduce as much and cause a severe infection in humans. Inactivated vaccines, you usually, same as live vaccines, tissue would be used to grow the virus, and then you'd isolate the virus, and then you'd either denature the virus through heat or through formaldehyde, and then if you use formaldehyde, you'd remove it, and you'd get a syringe and collect the vaccine. For toxoid vaccines, you'd use tissue to grow the bacteria, and then you'd isolate the toxins, and then you denature the toxins you'd inactivate the toxins through formaldehyde as well, and then you'd add aluminum salts to the inactivated toxins, and you'd collect the vaccine after removing formaldehyde. And with conjugate vaccines/subunit vaccines, you'd remove the viral DNA, you'd isolate it, and then you'd add it to a yeast culture, and you'd use a purifier to isolate it, the antigen, and then you would fill the syringe with the purified antigen, which cannot cause an infection. And recombining DNA and restriction enzymes, they're important because recombining DNA technology allows for splicing and fusing of DNA, and it can be used in making subunit and conjugate vaccines. It can be used for GMOs and protein-based medication. And restriction enzymes allow for certain DNA segments to be targeted in processes such as PCR or genetic engineering.
Edible vaccines basically convert edible food into potential vaccines in order to fight off pathogens. It involves the introduction of plants to specific desired genes, enabling them to make the desired proteins. Some advantages: it can be easily produced in mass level, and it's easier to take, and it has low risk of contamination. Some disadvantages is that the body can become tolerant to a protein or peptide, and people could ingest too much, which could be toxic, or they could ingest too little, which could cause an outbreak.
And influenza vaccine variability. One shot of influenza does not protect a person from year to year, since influenza is a constant evolving virus. Immunity to a single strand of influenza through infection or vaccination does not protect against new developing strands. A genetic material can change the amino acids that make up influenza virus proteins, which results in structural changes to the proteins that are properties of the virus.
Korea had an epidemiologist who monitored the health of the population, researched the patterns in the disease, and they examined the lifestyle factors and relations to chronic illnesses. The average salary range is between 24 to 110K per year, but it does depend on what state you're in.
Okay, so in conclusion, vaccines are important in day-to-day life as they help provide not only individual immunity but herd immunity. And with that, we've eliminated some very deadly diseases such as smallpox. And the immune system's collaboration with vaccines demonstrates the power of preventative care. There are many different types of vaccines, and they're constantly being built upon and improved even in the modern day. And concepts like epidemiology and herd immunity highlight the broader implication of vaccine production and the importance of vaccination in preserving our health. Any questions?
Alright, Gabby, what is the difference between an antibiotic and an antibody? Because I think those words were used while you were presenting part of it,
Yeah.
but it could have just been like a mispronunciation. So I just want to make sure you know this. Antibiotic, like what we talked about last semester, and antibody, like in the immune system. What's the difference between the two?
An antibiotic is a vaccine, or like it helps fight against a disease or bacteria. And an antibody, I guess it's a cell. Yeah.
So an antibiotic is like created and it fights off specifically bacteria. An antibody is created by our immune system and it fights off pathogens including viruses and bacteria. So make sure you know what the difference is. Vanya, can you provide an example of acquired immunity? But you're not good at that when it's not because you're talking about yourself.
So basically, it's like something that you get born with, not something that you're born with.
Any example would be sufficient.
The vaccination.
And the cars.
So you go into the doctor's office, they call your name, you get seated, and then a nurse or a nurse does it for me.
Attention scholars, can I
we'll
have
ask
the
you to
girls
roll up your
soccer
sleeve if you
team
have it.
report to
They'll wipe
classroom
it down with a
119
tissue-
at
with a
4:05?
alcohol,
And can I
and
have
then they'll
the
inject the
boys
needle,
soccer team
inject the
report to
vaccine.
classroom
And if there's 2 shots, they'll do
127
both.
at 4:05?
And then
Thank you.
put a Band-Aid on it, and you just put your sleeve back down, and you go on about your day.
Thank you very much. Appreciate that. All right, great job, guys.